All right, ladies and gentlemen, thank you very much for hanging on for this session on which has been very creatively titled. I think Bernie has to take credit for this. He's called it Humankind 2.0. I tend to try to be less dramatic about our work than that, but I suppose a lot of people do think of it as possibly the final frontier of medicine, the medical control of aging. And I'm going to give just some brief remarks before I introduce our distinguished panelists. One thing I should mention before any of you think about doing something else is that not only do we have the wonderful scientists who are listed on your program, namely Xianmin Zeng from the Buck Institute and John Cooke from Houston Methodist, we also have Mike West, who, of course, gave a talk just now upstairs during the previous session, but who will give an abbreviated version of it now. Mike is, of course, one of the absolutely world leaders in anti-aging research, and especially in industry, and has been associated with this conference almost as long as I have. It's certainly thanks to the vision of people like Mike and, of course, Bernie himself, that the combination of an intersection of gerontology with regenerative medicine has started to burgeon so productively over the past few years. So I'm just going to briefly outline the kind of work that we do at SENS Research Foundation in Mountain View, California. Essentially, we are starting from the realization that what you see on this slide is a misconception. This is the kind of classification that you might get if you ask people to describe the types of way that people get sick. They'll say that there are infections, in column 1, there are inherited diseases, the kind of things that a few of us get from our parents. There are the chronic and progressive diseases, predominantly of old age, that we see in column 3. And then way out in the stratosphere, there's this rather poorly defined, nebulous set of things that we think of as not really diseases at all, but that we classify as aging itself. And you might wonder why the things in column 3 have been so much more recalcitrant, so much more resistant to progress against from medicine than the things in column 1 and possibly even column 2. We've really done very well against almost all infectious diseases, at least in the industrialized world, but the chronic diseases of old age have of course risen to the top of the pile in terms of prevalence in the industrialized world, simply because of the lack of progress that has occurred. So we might ask, why is that? And most people would say, well, it's just because they're awfully complicated, they all happen at the same time, they interact with each other, it's just the complexity. But there is an alternative explanation, which arises when we start to think about what aging actually is. Aging is fundamentally a side effect of being alive in the first place. It arises from the fact that the body is set up to tolerate a certain amount, but only a certain amount of the damage that the body does to itself as a side effect of metabolism, of the processes that keep us alive from one day to the next. So for a long period of time, nothing much goes wrong in terms of physical and mental function as a result of the accumulation of this damage, but eventually there's too much of it and the pathologies of aging start to emerge. Now the thing is that that is a perfectly good description not only of what happens in column 4, but also of what happens in column 3 of this table. This table you're seeing now is the same as the one I showed you earlier, except that the huge black line is in a different place. And that is the misconception I was referring to. It's an extraordinarily damaging misconception, even though it may seem rather minor when I describe it in that way. And the reason it's damaging is because it has led to this. It has led to the overwhelming majority of the medical industry and the medical research community's attempts to do anything about column 3, being essentially to treat them as if they were like column 1, to treat them as if they were infectious diseases that could be eliminated from the body, which is of course nonsense once you realize that just like aging itself, they are side effects of being alive. You're not going to eliminate side effects of being alive from the body without eliminating being alive, which would rather defeat the object. So this is really what's wrong medicine is misdirected. Gerontologists have realized this. A few people have realized it for maybe 100 years. But the unfortunate effect is that the process that they have tried to emphasize has also failed to postpone the ill health of old age very much at all. The idea of most gerontology is to clean up metabolism, slowing down the rate at which damage is created, but it doesn't work, and this is why metabolism is just too complicated. This, of course, is a simplified diagram of a small subset of what we know about how the body works, and if you take into account the fact that it's dwarfed by all that we don't know about how the body works, then you can see pretty easily that it's fairly hopeless to try to actually tweak it, to stop it doing the thing we don't want it to do, namely the creation of damage, without at the same time stopping it from doing things we need it to do to keep us alive. So that's basically why the approach of slowing down the creation of damage has also not worked. At SENS Research Foundation, we pursue a third approach, which was pretty much ignored by the gerontology community until about 15 years ago when I started to promote it. And that is to go in and repair the various types of damage that the body creates, thereby essentially separating, uncoupling the process of damage creation from the process of the pathogenic effects of damage, and thereby allowing damage to be created at the natural rate without the eventual pathogenic consequences. This is how we go about it. We classify the various types of damage that the body does to itself into a relatively manageable number, 7 types of damage. These are broad categories, as you can see, but nevertheless they are useful categories. So if we look at the top, cell loss, simply cells dying and not being automatically replaced by the division of other cells. This, of course, leads to progressive depletion of the number of cells in the affected tissue or organ, with eventual consequences for that tissue or organ's function. And we know perfectly well what the repair strategy is for that, the maintenance approach. It is simply cell therapy, to put cells back that will divide and differentiate to replace the cells that the body is not replacing on its own. We're going to be hearing about later on in this session about how that kind of approach can be applied to Parkinson's disease, one of the most important aspects of aging that's mainly driven by cell loss. And by a bit coincidence, I see in the back of the room a professor that we're actually funding at SENS Research Foundation. He works at Wake Forest Institute on another of the major aspects of cell loss in aging, namely the loss of cellularity of the thymus, which is a big part of why the immune system doesn't work so well in the elderly. Now, if I had an hour, I would talk to you about all the other things down on this list, the various ways in which we are seeking to attack the other types of damage that contribute to human aging. Unfortunately, I don't, but suffice to say that the reason SENS Research Foundation exists is because there is so little work in most of these areas. Virtually nothing compared to the excellent work being done in stem cell research throughout the world that leads to the ability to have conferences like this. And, we really need to fix that, because essentially all of these types of damage are just as capable of killing us as loss of cells is, and without fixing all of them, we will not achieve, in my view, truly effective postponement of the ill health of old age. It's gratifying to see that this concept is finally, after more than a decade, reaching a level of acceptance, and indeed, I would say, even orthodoxy. Just a couple of years ago, a group of very distinguished grandees of gerontology decided to get together and write a long review in Cell. It was about 30 pages long, actually, which essentially reinvented the same idea that I've been describing in the last few minutes. They had 9 categories, but it was essentially the same classification, and they suggested, a divide and conquer strategy for repairing each of these types of damage. And this paper, rather unlike the one that I published in 2002, has been cited once a day ever since. So that's a very gratifying feature of this. Furthermore, our own work is moving forward quite nicely. Just 6 weeks ago, we had, for the first time, a paper published in Science magazine, which was from the work that we've been funding at Yale, looking at cross-linking leading to hypertension and other aspects, other consequences of the loss of elasticity of the extracellular matrix. We've had a few other successes that I mentioned there. I just thought, because of John Cooke's interest in the vasculature, I would very briefly mention the work that we've been doing at just across the street from him, actually, at Rice, looking at the reversal of atherosclerosis by equipping macrophages with the ability to break down oxidized cholesterol. We have identified enzymes in bacteria that are able to do this, and we've been able to modify them so that they work in human cells, with the result that we can increase very sharply the viability of cells exposed to certain otherwise lethal toxic concentrations of the main oxysterol that drives atherosclerosis, 7-ketocholesterol. This was published a few years ago, and for those of you who are more private sector people rather than academics, I should mention that this has now actually been moved into a startup that was founded by one of our major donors, specifically because he got so excited about this. That's all I've got time for, so I'll just mention that a few years ago I published this book which goes into gruesome detail about all the stuff that we think needs to be done and how we're doing it, and I hope that you will be able to get hold of it. And now I will start the session proper, so to speak, by introducing the panelists. We're going to start with Dr. West, because I think his remarks will probably set the stage for the presentations from the other two, at least to a greater extent than the other way.
So I wanted to give some background on regenerative medicine, on telomere biology for our next two speakers. You know, this is not a corporate slide deck. Earlier in the day, I gave a talk from BioTime. This is just my own point of view. This is my own perspectives. So the way we all look at aging differently. It's like the old metaphor of the people approaching the elephant, seeing different things. What I've always looked at life, rather than asking the question of why do we grow old, is that looking at the, thank you very much, looking at the immortal renewal of life itself. If you're a cell biologist like I am, or a stem cell biologist, the way we're in a reductionist approach to aging. And I like this picture because it highlights the biology I'm trying to explain to you. Is we're the result of a lineage of cells that have been proliferating for billions of years. The cells in our body will die for the first time in us in billions of years. And so, you know, what's so different about cells that branch off into the somatic cell lineages that cause us as an organism, as a multicellular organism, to have this aging phenomenon when the cells we were created from have no dead ancestors, unlike you and me. And so I think that's a very interesting thing, and it's basically what I'm going to talk about right now. So early on, you know, we came to recognize that there's a lineage of cells that branch off from this immortal germline. Is immortal, of course, meaning infinite. Anytime I use the word immortal, I'm referring to the infinite renewal or replication of cells. I'm not referring to any religious or science fiction type scenarios. So the germline has an infinite renewal capacity, but the cells that branch off and make the somatic cell lineages have a finite lifespan. And it's all the evolution of the genome over millions of years that changed the soma, and almost invariably giving it a mortal phenotype as opposed to the germline. So the way this became unlocked really was back in the 60s with Leonard Hayflick studying the senescence, or the finite replicative capacity or mortality of cultured cells. And what happened was, as we were trying to decipher all that, we came to a conclusion that it was linked to the biology of the telomere. And so this dichotomy that I just showed you of the germline versus soma is reflected here. Telomeres are maintained. You see it shows a wavy line there. We think there's actually some loss of telomeres and oocyte maturation, kind of a breathing, meaning up and down breathing of the telomere length in the germline, but maintenance through telomerase to allow the babies to be born young, simply put. But in somatic cell types, and you know, other than the germline, it might be all somatic cell types. Hematopoietic stem cells, certainly. MSCs, certainly. there's no telomerase. Hematopoietic stem cells I was equivocated a bit on because blood lineages that expand in the lymph nodes, like B and T cells, for instance, can reactivate telomerase. Telomerase was originally cloned from an inflamed tonsil. So in the germinal center, you have the clonal expansion of cells, and that really uses telomeres quickly, burns through the lifespan of cells quickly. So that's that rare conditionally telomerase positive arrow drawn on there. But most somatic cell lineages just repress telomerase activity and have a finite lifespan. Of course, that's reactivated in many malignancies, of course. So one idea early on that was pointed out by Aubrey was just transfer of that replicative immortality, or using telomerase to extend telomeres and then take telomerase away again, whatever variation on the theme that would be. The clue really that it was telomere attrition really came from two sources. Alexei Lovnikov, who was a Russian theoretician who dreamed up this wild idea out of nowhere. And then this data, which I saw in late 1980s, Howard Cooke published a paper on human telomere shortening in vivo. I don't know why people don't give Howard credit for this. This was the first published account of telomere shortening in human aging in vivo. And he showed that in sperm of men, as they aged, telomeres were not shortening. And that dichotomy you see there is what triggered me to start Geron and the belief that it was telomeres that was causing the Hayflick phenomenon. So we went after the components of telomerase to try to prove that. And as you may know, telomerase, if it's the immortalizing enzyme, it's sort of like the fates that weave the thread of life. It's a hand and fingers, thumb and fingers motif, and then it carries with it an RNA template that lays against the end of the telomere, and so it's a reverse transcriptase. It uses that RNA template to know how to make TTA-GGG on the ends of human telomeres. So it's template-mediated RNA, RNA-dependent DNA polymerase, so like a reverse transcriptase. So the idea was, can we use it therapeutically? The fun little anecdote, again, these aren't business slides, kind of fun intro slides, was the first human cells ever to see the telomerase gene exogenously added were from Leonard Hayflick himself. He was in our lab filming a documentary, and he handed me a piece of himself. It's a piece of his, for drama. Len's a dramatic guy. He hiked up his pant leg in front of a camera and chopped a piece of skin off his leg and handed it to me off camera, and that's his leg. And you can see the keratinocytes growing out and the fibroblasts. Anyway, we had his cells, and I was joking with him. I said, Len, we always talk about the Hayflick limit. We're going to really figure out what the real limit is. We'll track how many doublings your cells go. And we'll publish the Hayflick limit. And we were joking around, but then we had his cells and telomerase cloned and in an expression vector at the same time. And I thought, oh, this is too good. We've got to do this. So the first cells that were transfected were Len's cells. Then let me make another point. Aubrey put the metabolic pathways up there, talk about the complexity of human biology. Len is a dear friend of mine, wonderful guy. If you go back and look in the literature, as a responsible, he thought, scientist, and he is, he said numerous times, if he were here, if we were in a time machine, he was giving this little talk I'm giving, what he would have said is, that aging is infinitely complex, we'll never understand it, number one. Number two, the aging of fibroblasts, what we call the Hayflick phenomenon, is so complex. There's over 300 identified changes, everything from lipid transport to the structure of the nuclear envelope to all these things. He says, we'll never understand it, we'll never intervene in it, it's hopeless. He said that for years. He published on it for years. When one gene was added to his cells and immortalized his cells, I had the privilege of showing him the data in a restaurant one night. And he looked out the window and he said, it can't be that simple. And it was. I mean, it's a little complexity, some of you know. Cells can arrest under an oxidative stress that telomerase doesn't rescue and all that sort of thing. But the biology ended up being pretty doggone simple. So a lot of these concepts that we're laying out here, they almost sound too good to be true and everything, but some of this biology and all this complexity can be reduced to some very simple paradigms. One is life is naturally immortal in that you and I come from a lineage of cells that go back for billions of years. Another idea was, can we capture the germline cells and just use them? And that was the concept of regenerative medicine. So let's just grow and isolate human germline cells. And the idea was to get these ES cells or embryonic germ cells and make cells from them. And so what we showed was that we could capture the cells from these first few days of human development and expand them in the dish, and they're in an embryonic stasis. They propagate themselves. They're called embryonic stem cells, but they're really not a stem cell. They don't exist in you and me. There's not a niche that makes more embryonic stem cells or a differentiated cell. They're a cell that's in a stasis, that's in a very primitive state that has the ability to make all the cells in the human body. And they have telomerase. So this is the trap assay. On the left, you can see H9, one of the original embryonic stem cell lines, expressing telomerase. On the right, you see a whole bunch of cells made from them. And from the first 2 weeks of beginning to develop into the body, they turn off telomerase. So mortality begins, obviously, in the first few weeks of human development in utero. We've shown that we can capture cells at a primitive state. We call them embryonic progenitors that are also in a stasis, over 200 types, and isolate them clonally. And of course, as many of you know, the transplant rejection problem now has been addressed in multiple ways, one of which is nuclear transfer to make patient-specific cells, or IPS technology, taking a cell back to the germline. You see how all of these themes relate to the germline soma? Transporting cells back to the germline. So here's data from my collaborators published years ago that nuclear transfer can reset the Hayflick limit in the bovine species. Interesting. And then later on with IPS technologies and transcriptional reprogramming, you could reset telomeres back to the beginning of life. So the idea was, as Aubrey said, you could make your own cells that you were born from. And what's remarkable about this reprogramming, it hasn't been studied in a whole lot of contexts, but I think maybe some of our speakers might mention, it's been shown that it can not only reset telomere length, but can, recycle accumulated, lipofuscin, and can reverse a lot of the other changes that go on in aging. Having an immortal cell in culture, of course, allows us also to do multiple, even targeted genetic modifications, just like we do in the mouse. So we can make, for instance, genetic modifications, like has been published here, to make cells to escape immune surveillance, to make off-the-shelf allogeneic cells that are young of all kinds. And you can even do what some people have called regenomics. What the heck's that? Regenerative medicine and genomics merged together. So here on the right is my DNA in a Circos plot, and there's my arm skin cells, not lens, and my reprogrammed cells. And you could go in and say, Mike, I want to make, a genotype that's never existed in nature, to enhance human biology. And this is really, of course, the brave new world of the future of regenerative medicine. I want to make one other last quick thing, because I think this is an emerging theme which you'll be hearing more about in the future. Another kind of stasis in embryology is displayed by this animal. This is the Mexican salamander that many of you people know can regenerate even, profoundly, tissues like arms and legs, even the brain and eye and all these things. What is going on with these animals and many other similar animals is they're still in an embryonic pattern of gene expression. These animals normally would metamorphose into a land animal, like on the left, but instead they're stuck in a permanent state of embryonic development. It's called pedomorphism. And as a result, is they can do this profound regeneration. Well, it's possible to capture human cells the same way, in a pedomorphic state. Capture them in a permanent embryonic state. And what I'm showing you here, for the first time I'm showing today, some molecular markers of this. This is protocadherin beta 2 supercluster family. And the cells on the right are all cells that we've expanded in vitro of over 200 different kinds. And they are all showing this embryonic marker. And you can see on the left, normal adult cells of all kinds don't. And here's the opposite. Here's adult cell, an adult fetal and adult cell marker, and our cells don't express it because they're in this stasis. So what this means, in summary, is very complex ideas can be brought down to some very simple paradigms. Telomerase is a central regulator of the dichotomy of soma versus germline. We can capture cells in the germline stasis, which is IPS or ES cells, and use them potentially therapeutically. They're in the clinic. I gave a talk earlier today about some of that for macular degeneration and other things. And my last bullet point, and I think this is going to be an emerging theme in coming years, in coming meetings, by understanding the genes that are switched, just like telomerase was switched from germline and soma, but are switched in the transition of embryonic to fetal development, we could potentially unlock the regenerative potential that, like for instance, the Mexican cellular has in humans. And so we've called that induced tissue regeneration, and I think that's going to be a reality. And of course, if we could do that, it could profoundly have an impact on age-related degenerative diseases. So with that intro, you want to go to the next speaker?
So the question here today is, can human aging be reversed? And the definitive answer to that is yes. So the stigmata of aging, shortened telomeres, alterations in the nuclear matrix, epigenetic modifications, mitochondrial damage, excessive reactive oxygen signaling, deranged proteostasis, and reduced replicative capacity, all of that can be reversed. That is true at a cellular level. So that's where we are at the moment. Shinya Yamanaka, I'm sure you're familiar with him, won the Nobel Prize in 2012 for his generation of induced pluripotent stem cells and an understanding of the core pluripotency factors that play a role in the induction of pluripotency. He well deserved the Nobel Prize. Just 7 years after he published his first data, 6 years after he published his data, land speed record for getting the Nobel Prize. And that was because these cells, induced pluripotent stem cells, which you're probably very familiar with, are already changing medicine because these induced pluripotent stem cells can become any other cell. They can become any differentiated cell, endoderm, mesoderm, or ectodermal lineage, and can be used for understanding the pathobiology of disease, can be used for drug screening, can be potentially used for cell therapy. Now, these induced pluripotent stem cells are virtually all the stigmata of aging are reversed in these cells. And as Mike was telling you, telomerase may be playing a big role in this because telomerase is reactivated in the induced pluripotent stem cells. So you can take an aged fibroblast from a patient and induce pluripotency with those 4 factors, OCT4, SOX2, KLA4, c-MYC, 4 transcriptional factors that are necessary for pluripotency. You force the expression of those factors and you get something within about 3 weeks or so that is pluripotent. It's basically what you were when you were a twinkle in your mother's eye. A pluripotent cell that can become anything, in any tissue in your body. So human aging can be reversed at a cellular level, and a lot of this might be due to the reactivation of telomerase. Now, the same month that Yamanaka won the Nobel Prize, we published a paper in Cell that disclosed another limb of this pathway. So I mentioned to you that these genes, OCT4, SOX2, KLA4, and c-MYC, are absolutely critical for reprogramming. And this is what Yamanaka got the Nobel Prize for, his discovery of the importance of these transcriptional factors in inducing pluripotency. But what he did not know at the time, and what we serendipitously discovered, was that the retroviral vector itself was playing an important role in pluripotency. And what we found was that retroviral vectors or adenoviral vectors, whatever viral vectors people are using to force the expression of these genes, activates innate immune signaling, causing global changes in epigenetic modifiers that increase epigenetic plasticity to allow these factors to work. It was a serendipitous discovery because as a physician, I was trying to generate a way of making iPSCs that avoided integration, retrovirus. So we began to make cell-permeant peptides of the Yamanaka factors, OCT4, SOX2, KF4, c-MYC. And this is the team that helped me do it. And we made the factors. We showed that they could bind to their consensus sequence. We showed that they could get into the nucleus of the cells. They could even rescue iPSCs where we had knocked out one of the Yamanaka factors. So they worked, but we could not get them to reprogram fibroblasts, human somatic cells. And that failure led to the Cell paper, because in trying to understand the failure, we found a path. And that was a very interesting path. I'm showing you here one of the genes that's important in pluripotency. It's called NANOG. And this gene is turned on by SOX2, OCT4. It's one of the core pluripotency genes. And you can see what happens when we give retroviral SOX2 in red. We get an increase in expression of NANOG within a day. And with the cell-permeant peptide, we got nothing. And it's always wonderful when another postdoc in the laboratory can reproduce the work. This is the work of Jun Lee. This is the work of Nazish Syed using a different cell-permeant peptide, OCT4, or retroviral OCT4. And again, the retroviral expression of the Yamanaka factor increased NANOG expression, as you'd expect, but the cell-permeant peptide didn't do much. And then Nazish took this out for 21 days, and you can see that NANOG expression goes up dramatically, 50-fold with the retroviral OCT4, but only not sufficiently with the cell-permeant OCT4. He chose this time point, 21 days, 3 weeks, because this is the time point where we start to see the induced pluripotent stem cells pop up. So, for whatever reason, the cell-permeant peptides weren't working. And then it occurred to me that, based on some work I had done with Ed Makarsky at Stanford years ago, that the virus itself might be having an effect. So I asked the guys to throw in a retroviral vector encoding GFP, and then everything worked. We got the proper gene expression with the cell-permeant peptide, and we got iPS colonies. I won't go into all the data, but just this concept that, yes, the Yamanaka factors, OCT4, SOX2, KL4, c-MYC, these are absolutely critical for pluripotency, for reversing cellular aging. However, those transcriptional factors need an open chromatin configuration to act, because in the fibroblast, the very places where these transcriptional factors are supposed to be acting are locked down. The chromatin's not in an open configuration. And what we found was that activation of innate immunity through the toll-like receptor 3 causes global changes in epigenetic modifiers. The whole HDAC family is downregulated. Histone acetyltransferases are upregulated. And the cell is thrown into a state of epigenetic plasticity. Now, this phenomenon is important not just for nuclear reprogramming to pluripotency, but it's a primordial response that permits a cell to adapt to its challenges. So there are pathogen-associated molecular patterns that viruses and bacteria present. There are damage-associated molecular patterns that are uncovered by damage to the tissue. There are specific receptors for those patterns. Pattern recognition receptors like the toll-like receptors can recognize these molecular patterns and stimulate innate immunity. Well, that's well known. And inflammatory cytokines and chemokines are then released. But what we found was that this same pathway leads to epigenetic plasticity. And we now have data not yet published. We published this work showing changes in gene expression of epigenetic modifiers, but we now see post-translational modifications of these epigenetic modifiers that change their activity. The whole polycomb complex, or repressive complex, is repressed by innate immune activation. Why is this important to the discussion today? It's important because this process, which we found was important for generating iPSCs, induced pluripotent stem cells, can also be used, can also be manipulated in a therapeutic manner to change cell phenotype. So we published this paper this year in Circulation, an excellent heart journal, and we showed in this paper that we could take human scar cells, fibroblasts, and change them into endothelial cells, induced endothelial cells, with a small molecular approach. An innate immune activator, poly-IC, which is FDA approved as an immune adjuvant, and a little cocktail of EC transdifferentiation factors that, with outside-in signaling, allow the fibroblasts to transform into endothelial cells. And I won't go into all the data, but we show that these are authentic endothelial cells by RNA sequencing, by immunohistochemistry, by all the endothelial function studies. So this is a way of changing cell phenotype in a therapeutic way. How can this be utilized? Well, the clinical applications could include developing a small molecule cocktail for changing fibroblasts into vascular cells, for example. That's our interest is, as Aubrey told you. Now, if you have a patient that has a heart attack, what happens next, we try to minimize the damage, but what happens next is that cardiac fibroblasts migrate into that area of injury, and they proliferate and make extracellular matrix and form a scar. So what if you could modify some of those cells into endothelial cells that would self-assemble and form a vasculature that would provide perfusion and a niche for true tissue repair? So that's what we're working on now in small animal models. So this is the approach that we're taking. Now, I want to turn to another story that is related, and that is back to this question of age reversal and the role of the telomere. Yes, Aubrey. 5 minutes? 5 minutes? Okay. I'll be brief then. I think Mike West already gave a great introduction into the role of the telomere in extending in the Hayflick limit, and telomerase can reverse that telomere shortening that occurs with aging. We recently have used modified messenger RNA again to avoid integration of a gene into the human genome. Rather than using retroviral TERT, we're using modified messenger RNA TERT. And this took a little bit of tweaking to develop methodologies to get the RNA into the cell and the telomerase expressed in a functional manner. But it's working now. We published this with our colleagues at Stanford in FASEB this year. And basically what you're seeing here is in a normal human fibroblast, the Hayflick limit that Mike was talking about earlier here. After about 50 population doublings, the fibroblasts plateau. We treated the fibroblasts with 1, 2, or 3 treatments of the modified messenger RNA encoding telomerase, and we got increasing population doubling. And the interesting thing is that the cells do eventually plateau again because the telomerase is only transiently expressed and transiently extends telomeres, but then the cells continue to age. So we haven't transformed the cells. We have merely extended their lifespan. And what's interesting, this can be done again and again, and you can see after the cells become senescent, we can treat them again, and they can increase replicative capacity. Now, we're already applying this technology through our RNA core. We're making modified messenger RNA in our RNA core, which is funded by the National Heart, Lung, Blood Institute and the Cancer Prevention Research Institute of Texas. And we're making clinical grade RNA. In January, we'll have that up and running. We're making GLP RNA right now, and we're working with a company to develop a regenerative medicine therapy, improving their cell therapy using RNA telomerase to improve replicative capacity. And there's potential veterinary applications. We're working with the Progeria Foundation right now. We have funding from the Progeria Foundation to try to improve the replicative capacity of cells from these children, and we've been successful in doing that as one step toward a therapy for accelerated aging. And of course, progeria, the main problem is not a problem with telomerase. It's really a problem with a nuclear matrix protein called lamin A, and that abnormality causes distortions of the nuclear envelope and ultimately a shortening of the telomere that we've been able to reverse with the telomerase therapeutic RNA. And here's one slide to show you that we can increase the TERT expression in these cells. That's associated with a reduction in beta-gal expression. These are lines from two different patients. Patient 1 with a reduction in beta-gal staining, that's a senescence marker, and also the same thing seen in patient 2, and improvements in cell replicative capacity. This is a modest improvement in replicative capacity, but note this is over just a few hours. So we're following these for longer periods of time. The point is that we're now well on our way toward developing a new regenerative medicine approach based on work that Mike West has done and work that we did with Giron years ago, but now using RNA in a GMP fashion to develop a therapy to improve an adjunctive treatment to improve cell therapy. That's the low-hanging fruit, and ultimately, with systemic delivery, our nano engineers at the Research Institute, Mauro Ferrari, who's the CEO, and Ennio Tessiati, were developing methods to deliver the RNA systemically for these children with progeria. So thank you very much for your attention, and I look forward to the panel discussion.
Well, thank you very much, Aubrey. So I thought I'll just change my slides as Mike and John has talked a lot about aging and IPAs and EAs and all the aging-related mechanisms, telomerase and all that. So I thought I'm going to give you a case study of what I have been doing with using IPS-based cell therapy for Parkinson's disease, just to change the subject a little bit instead of going to the basic Parkinson's disease mechanism study. That's what I was planning to do. Okay, so cell therapy. So Parkinson's disease, just bear with me, give you a very brief introduction. So it's a degenerative disease involved in the death of dopaminergic neurons in the midbrain. So there is no curative therapy, but there are a lot of pharmaceutical treatments and also surgical procedures, including deep brain stimulation. So what I'm going to talk about today is to look at the potential of direct cell replacement therapy. That means you transplant dopaminergic neurons into the brain to try to restore the function. So the proof of concept of cell therapy for Parkinson's disease, it has been well established since the 1990s. 3 clinical trials with fetal transplantation has shown that several patients, or in each of the trials, they have shown significant improvement, but also there are problems with fetal transplantation. Many of the problems, which, for example, like the side effect of dyskinesia, which more and more evidence point out that is because of the preparation of the cell. Okay, so many of this and the limited material, this we think it can be addressed by alternative cell source. And in this case, I'm going to talk about human pluripotent stem cells, including both embryonic stem cells and induced pluripotent stem cells. Now, I'm not going to go to the details of how we produce the cells and the methods of generation, but I'm going to walk you through the steps of the strategy. So let me just point out one fact. Currently, cell therapy are regulated under the FDA tissue practice final rules. Okay, this is at least in the US. In Japan, you have the fast track, which you perhaps heard about the RPE trial, used autologous therapy last year in Riken. So in the US, clinic investigations with an unapproved drug must be conducted under IND. So here is our IND-enabling study strategy for EAC and iPSC-based cell replacement therapy for Parkinson's disease. So first, we have to source or generate PAC lines that are suitable for clinic use, and we should manufacture cGMP-compliant dopaminergic neurons for transplantation and perform preclinical safety and efficacy studies in suitable animal models and obtain the IND from the FDA to conduct phase 1 clinical trials. So start with sourcing the cGMP iPS cell lines, and I'm basically show the data in combination of both the iPSC and EAC derived cells. I can explain you the details if you want to know more. So here are the key steps of iPSC manufacture process. And I just want to point out the methods we use is to use the episomal vector, and the starting material is cord blood cells. So it goes through the tissue acquisition, cell isolation, reprogramming, and expansion and banking. So we have recently published a paper which we believe this is first the cGMP compliant iPS cell lines made here in the US, and this is really the work was done by the NIH Center for Regenerative Medicine, led by Dr. Mahendra Rao, and manufactured by Lonza, which I have been helping them with the manufacture and also reprogramming. So basically, here are some points. So NIH has commissioned 2 clones, and will make master cell bank from both clones available to investigators. So I have made the GAP reporter line that is in the safe harbor that we will also make it available to investigators just to do the preclinical studies. And we're currently writing a follow-up paper that we include the whole genome sequencing and SNP and chip data, HLA typing. All this we have based on our conversation with the FDA, which kind of comparability assays we need to provide for people to use as the starting material for manufacture cell therapeutic product. So the next step is to manufacture dopaminergic neurons for preclinical studies, and this is what we have done with the cGMP site, and which this particular data are from the manufacture we have done with City of Hope. So here is the process I have developed in the past 10 years, how to manufacture dopaminergic neurons from PAC cells. So I'm not going to go to the details, but I just want to point out one factor, the manufacture process that is start from iPAC or EAC, and then we make neural stem cells and make dopamine precursors, and this is the stage for transplantation. And we actually can cryopreserve the cells, and the cells can be used for transplantation directly without any further culture. And those are, the process has been transferred to a cGMP manufacture site, and particularly this is with City of Hope. So we have done 3 dry runs of the manufacture, and you can see the different lot with different lot size. And use these cells, we have conducted efficacy and safety studies, as well as the test for device comparability in large animal models. So here are a couple of the data that I'm going to show you with the studies we have done. And the first is the efficacy studies. And again, we have published this. This is, again, use the cells manufactured in the cGMP facility. And we have used the 6-hydroxydopamine model to test our cells for efficacy. Okay. Again, this is a model, is a common accepted model. You lesion one side of the striatum and then transplant the cells and then look at the behavior. and motor function improvement. So just to go through it very quickly. So the two control group is vehicle, and then the other group, the one marked the green, is the one that is just slightly before the product step, is 4 days before, which I want to make a point, you have to time it. So if you transplant the cells too early, they would not differentiate into the right cell type in vivo. If you transplant them too late, they are not going to survive, okay? So you can see the cryopreserved cells and the freshly preserved cells, they function both. And the study is lasted for 6 months, and you can see the amphetamine-induced rotation test that is significantly improved and persisted for 6 months. So these are the histologic analyses that show you, again, the control group, which you can see the survival of human cells, but you definitely don't see dopaminergic neurons, where is the case of the product. You see not only the human cells survive, they differentiate into dopaminergic neurons, and they actually make nice axons there. And this is another way to label the human cells with human antigen here. Again, these are all the evidence of the survival of the cells is the reason why the animal get improvement. So these are the additional dopaminergic or midbrain markers just to show you the survival of the graft. So we then also go ahead to test a device. This is a device developed by my collaborator at UCSF. It's a radically branch deploy system, which you can actually make one injection into 6 different sites in the putamen, which is important, as many people point out, how you're going to put the cell there that may have a consequence of side effect like dyskinesia. And this device can be integrated into a stereotactic platform, which has already been approved for using for deep brain stimulation. And this device, again, the platform can be monitored by real-time MRI. So we use our cells, again, in this case, we label with GAP, and then we transplant into the pig's striatum. So this is to test the device. We need to have the comparability data for the device in a large animal models. That's what the FDA told us. So this is look at the survive, and the paper, again, is published. So finally, we also have done the safety studies using these cells, and one of the studies we have performed is a 9-month tumorigenicity studies. We just use a Jackson lab as the CRO to do this. So in the interest of the time, I'm not going to go through all the data. So we had a pre-IND meeting earlier this year with the FDA, with the data I have presented to you. So, and we have basically received positive comment, at least for the manufacture and for the clinical design part. So here's where I am with the Parkinson's disease cell therapy. We have access to clinical-grade IPS lines, and we have identified the manufacture site and access to the drug master file, and have performed preclinical studies with cells manufactured by the cGMP compliant process. And we have also coordinated with clinicians on surgical trial design and had a pre-IND meeting with the FDA. And at this moment, we are seeking funding to manufacture cells for phase 1 trial. With that, I want to thank the people who have helped me.
Yeah, thank you for that great question. We do have evidence that other receptors can have this effect. I think it's, and also RIG1, TLR4. So we do have evidence that other pattern recognition receptors have the same effect. Yeah, I'm sorry I didn't have time to go over all of that, but yes, NF-kappa B, IRF3, IRF7. So the typical kinds of things you'd expect from innate immune signaling also activate these changes in epigenetic modifier. Well, we haven't tested that one specifically, but we have tested modified messenger RNA, retroviral approaches, even the cell-permeant peptides, integrated cassettes. They all rely upon activation of innate immunity to induce pluripotency. You need to have epigenetic plasticity for those factors to work. So, we haven't tested that particular example, but in the 4 examples I've just given you, innate immune activation is required.
Any other questions for any of us? Go ahead. One about HIF1. Well, okay, so HIF1 is activated in all manner of circumstances, and certainly it has a large role, a diverse role to play in regulating other genes. We have steered away from that kind of thing in what my foundation does, largely because of what I said in my remarks about the complexity of metabolism. We believe that this is a classic example of, HIF1 is regulated really in a really sophisticated way for a bunch of good reasons. If you try to mess with the way it's regulated in the context of profound ignorance of the way biology really works, then the chances are very high that you will do more harm than good. But, of course, we love to be proved wrong about things like that. We love it if people actually demonstrate that things are working.
So the question is, if telomerase is being used as a therapy, could it increase the risk of cancer? Is it what you're asking? And certainly, probably 90-95% of cancers have a reactivation of telomerase, and those that don't have a reactivation of telomerase have an activation of this alternative approach to telomere lengthening, and that's what allows the cancer cells to continue to replicate. This is why we've chosen to use a non-integrating strategy to increase telomerase activity just for a short period of time. And I don't have any evidence that that would increase the risk for cancer, but I'd be very interested in what Mike West has to say.
Is that one working? I know this one is. I think of it, since we only have a couple of minutes, as a highway that gets narrower as you go down, with cliffs on both sides. So there's aging and cancer. And it's an easy road to walk when we're young, and if we extend human lifespan, cancer is going to become more and more of a challenge to manage, and it's going to be a tightrope walk eventually. And yeah, just as John said, the transient expression of telomerase, even systemically, you would not think that it was probably, how they do these cancer studies and saying eating bacon has a 3% higher risk of malignancy or something. I mean, it'd be something like that. You'd have a higher risk of malignancy, but the longer constitutive expression of telomerase, I mean, now we're guessing, but based on all the animal data, if you immortalized every cell in the human body, you probably would come down with cancer. An adult would probably, an adult would probably come down with malignant cancer probably within a year or two. But transient expression is probably a part of that tightrope walk. Does that make sense?
So yeah, I was delighted that John mentioned alt, because actually that's an area where we have a big project, and the reason we do is because hardly anyone works on it. It was discovered 20 years ago, and yet lots of people have looked at it briefly and found that it's really hard to figure out what's going on, and they've gone away and done something easier. But it's crazy because people are developing anti-telomerase therapies. Of course, the leader in this area is Mike's old company, Geron, doing a clinical trial in this area. But of course, you would predict that once that really gets to work as an anti-cancer therapy, it's going to select for alt, right? So alt is actually being underappreciated by a huge amount in our view. About the question you asked, though, I'm actually a little less optimistic than my colleagues here about the ability of transient activation of telomerase to avoid, to minimize the cancer risk. It seems to me that if the activation is ubiquitous across, is systemic, then it's going to help the cancer cell as much as it helps any other cell. And so the question is, how much do you need to help the other cells? And it may be that you help the cancer cells too much anyway. So it is unclear. But then there is also a big controversy about whether activating telomerase will actually facilitate cancer anyway. There are some prominent people in this field, including another person who used to work closely with Mike, Bill Andrews, who's now very prominent in this area in the public eye as well, who say that there's a good chance that telomerase activation may be protective against cancer simply because maintaining telomere length at a respectable size will avoid the important point of crisis that causes the huge genomic instability required to turn telomerase on in the first place. I'm kind of with the majority who think that it will actually be bad for us in terms of cancer, but I think there's still a lot to be found out. Yeah, the biggest progress is money, and the solution is for someone in this audience to give me large amounts of it. And I wish I were joking, but the fact is that when I started getting interested in doing something about aging 20 years ago, I realized that I had three problems. Number one, I didn't know how. Number two, I didn't have the people who were interested in helping me to do it and who had the expertise I didn't have. And number three, I didn't have any ability to pay them. And I was quite gratified at the ease with which steps 1 and 2 fell into place, but step 3 is still a big hole. And it's completely astonishing, and of course this applies whether in the academic setting or in the non-governmental setting such as SENS Research Foundation, or in the private sector where Mike works. None of us have anywhere near the funding that we would need. I estimate that our work is probably being held back by about a factor of 3 in terms of the speed it's going, purely because we are so strapped for cash.
For RNA therapeutics, the major problem, Graham, is delivery. How do you deliver the RNA to every cell in the body, and how do you protect it from RNases, which are ubiquitous? So we're taking a nanoparticle strategy to address that. But we're also going after the low-hanging fruit. We're doing telomere extension in isolated cells to improve other people's cell therapies. So we're going to use that as a first approach toward getting knowledge about telomerase, getting it into the clinic. That's how we'll do it, using ex vivo administration, where we don't have to worry so much about the delivery issues.
I would agree with the comment about people. We just need, I keep thinking of President Kennedy saying, in this decade we'll go to the moon, but then they poured national resources into the project. Can you imagine trying a project that had technological barriers like they had, going to the moon and coming back? Trying to do it on a shoestring like we do with small biotech companies and all that. We have a National Institute on Aging, but it's dramatically underfunded. We have a tsunami coming our shores with the aging boomers in the U.S. and outside the U.S. The numbers would scare the pants off from you. I mean, trillions of dollars of healthcare costs through Medicare coming to our shores. You'd think the United States would mobilize through NIH really significant increases in funding, because it's really scientists and funding and people. As Xianmin, you mentioned for your project, right? I mean, you really need the people and the resources.
Yes, to continue with what Mike said, I also think, for extending lifespan, I would more say to extend health span. So if we can use the resource that we could put together to, look at small molecules or other drugs that can use as an intervention to prevent instead of you get sick and then cure. So if we can extend that lifespan, which I think regenerative medicine like IPAs and other cells provide a tremendous tool for that. I didn't talk about that today because I was using a cell therapy as a case study, but I think that's the most difficult part to overcome, how we are going to do it together, how we are going to pool the resource, have some standard, and then not only talk about it, act on it.
Yeah, but that was always, back in the Geron days, I don't think I'm violating confidentialities, we modeled all that with computer programs. And so, I mean, it was always clear that if you people did years ago, they did clonal expansions of cells from different aged people, and they didn't look at the lifespan of the whole culture, but they looked at the lifespan of individual cells within an explant from a tissue, and they looked at all that sort of thing. And it was always clear that the vast majority of cells in the human body have way less doubling potentials that would allow multiple genomic hits. And so, like you say, the first or the second genomic hit has to be the activation of telomerase. Otherwise, the cells can't have enough numbers to have statistically probable second hits or third hits. So it was always clear very on that multiple, if you guys look at your skin or could look in your intestines or your brain or whatever, you would see pre-malignant lesions. I bet everyone in the room, because we're all relatively old adults, we all have pre-malignant lesions, right, that have undergone 1 or 2 genetic hits. Moles and things have started to grow and stop because of the lack of telomerase clonal expansions. And so that doesn't surprise me. And we were talking about the idea that telomere capping with telomerase could help prevent malignancy, and that's a flip side of the coin. That's probably true, but I just shake my head because all the best data suggests that if we constitutively immortalized all the cells in your body, that would unlock all these pre-malignant lesions, and you immediately progress into more and more genetic hits and/or progression of the disease. So we probably have. I think I once heard we probably have something less than 100, but more than 10 pre-malignant lesions just waiting for telomerase in our bodies.